Dr. Madhav Reddy, Principal, GPRCP, Dr. Prabhashankar, President, IPA Telangana State Branch, and all the registered faculty delegates. Good morning all. On behalf of management and staff of G. Pulla Reddy College of Pharmacy, I, Jayanti Reddy, Associate Professor, Department of Pharmacology, warmly welcome you all to the e-FDP on Insights into Drug Discovery and Development, IDDD 2020, organized by the Department of Pharmacology, Pharmaceutical Chemistry and Pharmacognosy, GPRCP, in association with IPA Telangana State Branch and MYBO Group. Drug discovery and development are among the most important translational science activities that contribute to human health and well-being. Your safety is our concern during this pandemic time. With this view, we are organizing the third in series e-FDP to help the faculty learn the innovative methods of drug discovery and development. Let us pray to the Almighty, help us come through this turbulent time and guide a pharma fraternity to bring out a solution at the earliest. We seek the blessings by lighting the lamp and a prayer song. I invite Archana Reddy, Associate Professor, GPRCP, for the prayer song. Welcome, Archana.
Good morning, one and all. Myself, K. Archana Reddy, working as an assistant professor in G. Pulla Reddy College of Pharmacy. Vandanam, Vandanam, Girinandini, Priyanandana. Vandanam, Vandanam, Muni Brunda Hrudaya Syandana. Vandanam, Karivadana Karuna, Sadanani Pada Kamala Mulakada Vandanam. Vandanam, Vandanam, Girinandini, Priyanandana. Vandanam, Vandanam, Muni Brunda Hrudaya Syandana, Muni Brunda Hrudaya Syandana, Muni Brunda Hrudaya Syandana. Thank you.
Thank you, Archana. I invite our principal and convener of this program, Dr. Madhav Reddy, to give his welcome note. Welcome, sir. Sorry, there is a technical issue. So I invite Dr. Viresh, Organizing Secretary of Department of Pharmacology, to speak. There's another technical issue. I'm extremely sorry for that. Now I invite Dr. Sama Venkatesh, Professor, Department of Pharmacognosy and Organizing Secretary, to address the audience. Welcome, Dr. Venkatesh.
Thank you, Reddy. I hope you are all able to listen me. Fine. This is the second part of the e-FDP program from the Pulla Reddy College of Pharmacy. And the first one is grand success, and nearly about 1,000 participants has been taken place. And the second part is a main theme for the present scenario, the drug discovery process. The three basic departments, the cognosy, ecology, and chemistry departments, combinedly hosting this e-FDP program. I request all the students, research scholars, and teachers make use of this FDP program. And this present scenario, drug discovery plays a very, very important role in invention of the newer drugs to fight against the pandemic diseases. I congratulate all my colleagues, and we are very thankful to the principal giving consent to conduct this program immediately when we put forward the proposal to organize this e-FDP program. All the speakers which have been identified are eminent speakers in their respective fields and having vast experience. And they will be putting forward their experiences and ideas. And I request all the participants to make use of this program very well and actively participate in the question and answers also. I once again thank Mrs. Jayanti Reddy for making me to present myself before you. And also, I congratulate Dr. Naresh and Bhasha who made this program, live program, and a pleasant one. And again, I congratulate each and everyone who has taken part in this program and making this program successful. So drug discovery process is very, very important present scenario. People are running behind the inventions. Invention is the major criteria, no doubt, but exploring the hidden knowledge which is there in the science and technology is very, very important. Please try to understand there is a lot of difference between the discovery and invention. Discovery, a drug discovery is a very big area to understand and require a logical thinking and application of the various fields. That is why the three departments combinedly hosting this, as these three departments are the basic departments in the discovery of the drugs and newer molecules. I wish everyone to make use of this program very well and actively participate in the question and answers and make use of this program is very well. And in this pandemic situation, the people can gain more and more knowledge, and this is the best opportunity for us to have a pleasant interaction between with the speakers. And I wish the program very, very successful. Thank you. Thank you very much.
Thank you, sir. Now I invite another organizing secretary, Dr. Videsh, Professor, Department of Pharmacology, to introduce the department. Dr. Videsh.
Yeah, thank you, madam. Good morning, all of you, and warm welcome for this IDD 2020 program, FD program. Um, uh, Most of the objective is just to were to bring what are the techniques for the research process. And actually, before COVID pandemic, we have planned to conduct hands-on training program, because already in Department of Pharmacology we completed one FDP hands-on training in the year 2018. With the feedback from the first FDP, we thought to conduct one more in support of our pharmacology faculty. And due to unavoidable situations, and we made to organize one e-FDP program. Shortly, we are going to conduct the hands-on training with support of our principals and management. First of all, I am very much thankful to management and principal sir. Main special thanks to principal sir due to his encouragement and being a department pharmacology professor, and whatever development happens in my department due to encouragement principal sir. Recently means we completed various collaborative work with Usman University, Mysore University, IIT Hyderabad campus. Recently we completed 4 projects, means 4 collaborative research work from IIT Hyderabad campus, and they are also happy with that, means whatever the service we provided to them, and even though they are going to continue same support from our side. And meanwhile, I found in the morning nearly more than 250 pharmacology staff are registered for this one. And whatever the speakers we choose, apart from their theoretical background, they're expert in practical also. Means they can demonstrate whatever the skill they know, which today they are presenting, and very good enough. Even though we can avail the whatever skill from them, and you are utilized promptly, and you can share. Meanwhile, if you have any pharmacology experts is there, then if you have interest to show your any skills, then bring to my notice, then we'll make it some hands-on training program by utilizing your service. And I'm very much thankful our Naresh sir and Nasib Basha, means what arrangement they made, because we are not good enough with the ICT tools, but with support of Naresh sir and Nasib Basha, and we can able to convey the meeting, means from FTP smoothly. Thank you one and all. Thank you.
Thank you, Dr. Virish. It's good to hear that a lot of participants have joined us. Now, I invite Dr. Naresh, Associate Professor, Department of Pharmaceutical Chemistry and Organizing Secretary, to speak about the department. Dr. Naresh.
Thank you, madam. I'm Dr. Naresh, Associate Professor, Department of Pharmaceutical Chemistry. On behalf of our department, I welcome you all to this e-FDP, Insight into the Drug Discovery and Development IDDD 2020. About our department, Department of Pharmaceutical Chemistry is established in the year 2003. We are actively involved in teaching and research while adopting new technologies and advancement in the field of drug discovery and development. Our labs are fully equipped with all the facilities to support research in synthetic and computational chemistry as well. Till date, total 15 MPharm batches successfully completed from the department, and students are placed in reputed pharmaceutical industries, research institutes, and government organizations. So with this short introduction, now I thank one and all those who have participated in this e-FDP program, and I wish everyone a happy learning in this e-FDP program. Thank you, madam. Over to Jayanti, madam.
Let me also quickly introduce our organizing committee members to you. Dr. Radhika, Mrs. Sita Mahalakshmi, Dr. Shravanti, and Dr. Nagesh are from Department of Pharmaceutical Chemistry. And from the Department of Pharmacognosy, we have Dr. Ravi Kiran, Mr. Ravi Kumar, and Mrs. Rajeshwari. From the Department of Pharmacology are Dr. Lingesh, Mr. Sri Hari, Dr. Lalita Devi, Dr. Padmavati, and me, Jayanti Reddy. Moving forward, now it's time for the scientific session to begin. I invite Dr. Padmavati, Associate Professor, Department of Pharmacology, to take charge of the session. Welcome, Dr. Padmavati.
Thank you, madam. Thank you, Jayanti, madam. Good morning, everyone. So before begin the session, there is an announcement. There is a small change in today's program. As per the schedule, we have 2 sessions today. So because of speaker's busy schedule, the first session on preclinical requirements for IND submission is rescheduled on 1st August. That is on Saturday. So with this announcement, let us begin the session. Good morning, everyone, once again. On behalf of myself, Dr. R. Padmavati, Associate Professor, Department of Pharmacology, on behalf of G. Pulla Reddy College of Pharmacy, inviting you all to the first day session. There is no limit for learning. So being teachers, continuous learning is our first priority. In this regard, G. Pulla Reddy College of Pharmacy is organizing continuous series of e-faculty development programs to enrich the faculty in various disciplines of pharmacy. As part of this, today we have an eminent and knowledgeable teacher to enlighten us on the topic of simple guide to immunohistochemistry methods and protocols. So let us welcome today's speaker, Dr. Lakshman Puli, Assistant Professor, School of Pharmacy and Technology Management, SVKMs and NMIMS, deemed to be University, Hyderabad. Welcome, Dr. Lakshman, sir.
Hello. Good morning, everyone.
Thank you, sir. So now I invite Dr. A. Lalita Devi, Associate Professor, Department of Pharmacology, to introduce Dr. Lakshman.
Thank you, madam. Good morning, one and all. I welcome you all for 1 week e-FDP program on Insights into Drug Discovery and Development 2020, organized by G. Pulla Reddy College of Pharmacy, Hyderabad, in association with Indian Pharmaceutical Association, Telangana State Branch, and MYBO Group. It gives me immense pleasure to introduce today's speaker, Dr. Lakshman Puli. Dr. Lakshman Puli is an experienced biomedical professional who has more than 10 years of research experience with specialization in neuroscience pharmacology and animal models. He has worked in reputed research institutions across Europe, the United States, and India. Dr. Lakshman Puli is currently working as Assistant Professor at School of Pharmacy and Technology Management, SVKMS, NMIMS, deemed to be University, Hyderabad. He got his PhD from University of Eastern Finland. He received his postdoctoral training from Polyclonal Medical College, New York City, USA, and Karolinska Institute, Sweden. His main research focus has been on brain immune responses and animal models of Alzheimer's disease. His main contribution to the scientific world include efficacy trials of intravenous immunoglobulins in mouse model of Alzheimer's disease. His work has been published in reputed international journals and presented in several international conferences. He was the recipient of Leon Levy Neuroscience Fellowship and North Savo Regional Grant of Finnish Cultural Foundation. He also received the European Union's prestigious Marie Curie Early Stage Training Fellowship. Dr. Puli aims to harness his scientific knowledge in pursuit of simple, economically viable solutions for societal problems that are existing within therapeutic and diagnostic domains. With this brief introduction, I welcome Dr. Lakshman Puli to continue with his talk on Simple Guide to Brain Immunohistochemistry Methods and Protocols. I welcome you, sir. Over to you, sir.
Hi, my voice is clear. Can you hear me? Hello. Can you hear me?
Yes, sir. Your voice is clear, sir.
Yeah. Very good morning to all of you. Very good morning to all of you, and thank you, Lalita Madam, for a wonderful introduction. It's been my pleasure, and I thank the organizing committee, our principal sir, Professor Madhav Reddy, Professor Viresh Mental, and the organizing committee of this e-FDP for providing me this wonderful opportunity to share my expertise with all of you. Unfortunately, because of this coronavirus, we have been reduced only for this online experience. Otherwise, we have agreed previously to have teach you some techniques online, but then again, we'll try to make best use of it. So, yeah, thank you very much, and I think we should get over with our topic for today. And I think I'll start my presentation. Let me share my screen. Please let me know someone if my screen is visible. So my screen is visible?
Yes, sir. Please keep in presentation.
Yeah, it's now in presentation now. All right, so the topic I have been given was a simple guide to brain immunohistochemistry methods and protocols. When we first discussed with Professor Viresh, I was told that I have to share a research protocol or a technique which most of the students here in Hyderabad can use it in their master's as well as PhD projects. So initially we thought that we'll give them a simple staining protocol and then an image analysis technique. But then again, I will try to do justice by giving everything on our presentation. And the title, as you can see, it's a simple guide, and don't take it as a comprehensive one or a complete one. So brain immunohistochemistry, it's a technique which anyone can adapt. It's a very simple technique, but then again, it is also written with lots of caveats and limitations. So you'll have to be really wise enough to choose which method, which way you have to go, which format you have to go. I will touch upon with about all of those. So the main objective of this presentation is to give you some of the basic science behind this immunohistochemistry, and then how to actually design a immunohistochemistry protocol, and then what are the opportunities, what are the choices you have got, and in what way you will choose the best one, and those kind of things. And then I will also walk you through workflow, and then at the end I will give you some examples how I have used all these principles and managed with some examples from mouse brains as well as some post-mortem human brain samples. So with these objectives, let us move on and let's see. So what is immunohistochemistry? I mean basically the term immunohistochemistry, if you break it down, immuno means anything that is related to immune system, and more specifically here immune system, I mean antibodies. So histochemistry, histo means tissue, chemistry is chemistry. So the chemistry in the tissue initiated by the antibodies, you can think of it in such a way. So what happens is that immunohistochemistry is a technique wherein which not only you will be able to identify whether there is a protein of your interest or not in your sample, but also where it is present. So basically immunohistology is a very specialized, it not only gives the presence or absence, but also gives you whether, it gives you the three-dimensional or the contextual reference to that protein, for example, where the protein is present inside the brain. So as a neuropharmacologist, I am mostly interested in studying the brain structure and function, and I have used this technique successfully for the past 10 years, and it has been an amazing journey so far, and I liked it. And also one way to learn immunohistochemistry is to enjoy doing it, and of course it is not going to be done in a day or two, but then as you practice it gets better and better. So the basic idea, as I said here, is to use antibodies to identify proteins in the tissue, thereby giving us the information where the protein is present, in what circumstances that protein is present. It thereby gives us vital details about the physiology and pathophysiology of the disease itself. So immunohistochemistry again, it's a very specific technique. I mean, it has got its own window for specificity and sensitivity, and the best part that I like with immunohistochemistry, it's ease of use. It doesn't require any hi-fi equipment to incorporate. It's very basic instruments you need. And then another nice thing about immunohistochemistry is it is very flexible. That means that you can use the antibodies, which are the main workhorses of immunohistochemistry, and you can adapt it to your own use. For example, you can tag the antibodies with an enzyme or with a fluorescent dye, or with you can use the same antibodies for in a column also, affinity columns also. So the ease of adaptability and the ease of use and a very broad specificity, not broad specificity, again, it depends upon the kind of antibody use, the sensitivity as well as the ability to localize the structure as well as the function. That is what makes immunohistochemistry an ideal tool for researchers, especially when you're dealing with, if for example, you have a new drug and you want to know what sort of effects or impact does it has on neurons, then probably it is best, and you know that it has got a very specific effect on a group of neurons, then you can use this technique and see whether these neurons are doing good or doing bad, whether there is there are healthy neurons are present, or the neurons are sick and they require any attention. So not only that gives about the qualitative aspects of the tissue, but also recent advances have allowed us to know also the quantitative aspects. Now, when I say quantitative aspects, it's not an absolute quantification, but then you can get a reasonable semi-quantitative quantitative assertion can be made using the techniques of image analysis, which can be clubbed along with the same immunohistochemistry line. So in this presentation, I would also give you a glimpse of it. So let's move on and see how it goes. So basically, immunohistochemistry, as you see, it's a combination of the immunology field, wherein which we have taken antibodies, and then you have histology, means the tissue study of the tissue structure, and then the biochemistry involved in how the immunology is able to bind to this target protein, and then all of these. After this, the most important one is microscopy, which actually magnifies what's happening in miniature scale. So all these, the conglomeration of all these different fields is what is a beautiful immunohistochemistry, and we're going to see how it is going to be. Before we go, it just makes sense to know about who are the pioneers of this immunohistochemistry. Among them, I think you all are aware of Paul Ehrlich. He is the guy who received a Nobel Prize in 1908 for his important concepts of immunity. So basically, he's the person who actually described the interactions of an antigen and an antibody. Now, thanks to COVID, everybody knows what an antibody is. Antibody is a protein molecules that your body produces in response to an infectious agent, which in case of COVID is a coronavirus. However, this presentation has got nothing to do with the coronavirus, but we do incorporate lots of those antibodies, the concept of antibodies, and these basic concepts were given by Paul Ehrlich. So he first described how the antigen and antibody interact with each other, what are the forces involved, and he made the preliminary observations, and because of which the entire field is standing on. Even the basic tenets of the pharmacodynamics is also because of Paul Ehrlich's work, and so rightfully in 1908 he got the Nobel Prize. But then if there is a father of immunohistochemistry, if you want to have a specific person who is responsible for it, that should be Albert Kuhns. So Albert Kuhns in 1940, he first used, he was actually studying immunology, and he was working with antibodies, and then an idea struck to him: if an antibody can bind with an antigen, why not use an antibody to identify a protein in a tissue? It's a completely out-of-box idea, and he reported it in 1940 and 41. But then again, shortly after that, he went for a military training during those times. And then again, by the time it really became evident, then it was already 1950s. But then again, the initial use of antibodies for identifying the tissue proteins, even though it's on 1940, by the time it was adapted in the neurosciences, it was only in early 1970s. So it took really a lot of time. It almost 20 years time it took. So it always, but then again, we can't blame those times. I mean, they don't have at that point of time, they don't have the kind of a network what we have today. So but anyway, by using this immunohistochemistry technique, the neuroscience field itself has progressed tremendously. It has led to the identification of different types of neurons. It has led to the identification of different functions. It has led to the identification of definite brain regions specific for function, not only in terms of physiology of the brain, but also pathophysiology. It has also led to the identification of protein deposits inside the brain. I myself am interested in studying about the deposition of proteins inside the brain, namely Alzheimer's. In Alzheimer's, there is this amyloid beta protein that gets deposited. So again, wherever there is a protein, and wherever, if you have an antibody, you can simply employ this immunohistochemistry to find out not only the presence, but also give you the contextual reference where it is present, what type of neuron it is present, what sort of shapes and what sort of contextual information it can provide a tremendous information about that. So thanks to Mr. Albert Kuhns, and the main experiments, what he did was he tagged these antibodies with the fluorescent tags, and then he threw it on the tissue, and upon observing under fluorescent microscope, he could see that specific regions of the tissue were lighting up, indicating the presence of antibodies. So that was the very first application of these antibodies apart from the therapeutic and diagnostic approaches. So as a concept, it's simple, straightforward concept. I mean, our body is made up of full of proteins. I mean, there is protein everywhere. So is the case of brain. So when there is a protein, then if there is a protein, then there could be an antibody also. You can develop antibodies for literally any protein. You name it, you can give it. And especially in days like these, you can develop monoclonal antibodies or polyclonal antibodies in a very short span of time. So when you have these antibodies for a specific protein, and when these antigens and antibodies, they are brought into close proximity, then what happens is that these antigen antibody, they bind together, and the very strong van der Waals forces of attraction keeps this complex stable. And once the complex is stable, then this can be used as a target for the secondary antibody, which is capable of detecting the primary antibody. The only difference is that the secondary antibody now has some kind of a tag. It could be a fluorescent tag, it could be an enzyme, it could be a gold nanoparticle, you name it. So once it has a tag, it can, this particular place where this target protein is present, it can be identified. So once it is identified, then it could be actually monitored and it could be studied. That's what it is shown here in this scheme. So you have this primary antibody detecting the antigen, and then you have another antibody which is tagged with an enzyme which is detecting the primary antibody. And this again, when a suitable substrate is given, this enzyme converts a soluble substance into insoluble signal. This insoluble signal can be actually be observed under a microscope, visualized under a microscope, and then you can say yes, there is a presence of protein or not. So essentially, immunohistochemistry answers only two questions: whether a protein is present, yes or no, kind of to the basic. It tells yes or no kind of a answer. In terms of quantification, it is very difficult to quantify the amount, because for quantification, you all know, we all go for ELISAs. ELISAs is absolute, it's in solution form, and you can really quantify how much of the target antigen is present. But however, here that kind of an absolute quantification is not possible here. At best, you can say is a semi-quantification. But then again, for a researcher like me, I mean, semi-quantification is also a good deal for me. I can get a good idea to what extent a protein is being expressed, and that will provide a lot of information for me itself. I don't need to work with absolute values. So just because the conversation is going too much about the antibodies, it's very much I mean, obvious to study what are antibodies. Antibodies are nothing but the proteins, also called as immunoglobulins. They are simplified by the word Ig. So as you can see, they are Y-shaped structures, and if you really look at it, it has got a heavy chain, this one, and two light chains, right? And there are plenty of these disulfide linkages. These disulfide linkages, they bring together the light chains as well as the heavy chains of an antibody together, and functionally they work as one single antibody molecules. So there are different types of antibodies are present in your body. There are different 5 classes of antibodies are there, and within a class there are subclasses of antibodies are present. And as a immunohistochemist, I'm mostly interested in IgGs and basically, and also IgY. IgY is also a kind of antibody. It is a chicken antibody. So I will tell you why chicken antibodies are also very interesting. So in terms of what are the functions of this antibody, there are 2 main functions of these antibodies. One is the binding of the antigen. The other one is the effector portion. So this region, which is called as a FAB region, this is responsible for the interaction that I was mentioning, that this region, FAB region, is responsible for binding to the antigen, whereas the FC portion of the antibody is responsible for effector functions. Now, what are effector functions? Effector functions means in our body, antibodies, once they bind to the target, there has to be some function. So it has got 2 functions. One is binding function and is functional effector function. The binding function is okay. Let's try to discuss this with the help of a coronavirus. So if I say I have an antibody for coronavirus, the antibody will have 2 functions. One is binding function. So binding function means the FAB region of this particular antibody will bind to the coronavirus. Then effector function is because of the FC region. This effector region will be responsible for mounting immune response against the coronavirus. So the same happens in the case of immunohistochemistry also. There is a binding region and there is an effector region, and then the binding region is responsible for identifying the antigen in the tissue, and then effector response is responsible for its effector. But then clearly we know that in immunohistochemistry there is no effector function. We don't want them to do any effector function because it's a test system, right? It's not a living system. So essentially we don't really need FC region, but then again getting rid of FC region itself is a task, so there is nothing problem in leaving it like that. But then there are specific types of antibodies like camelid antibodies where I mean it has been miniaturized. Those limitations has been resolved, but then again we'll consider for all reasons that is a Y-shaped structure with FAB and a FC region. So as I already told you that the FAB region is responsible for binding the antigen. Here in this scheme, the antigen is a infectious agent, and hence this FAB region is binding to it, and then the FC portion is responsible for its effector function. Here the effectors are eosinophils and macrophages, even antigen presenting cells like dendritic cells. So basically antibodies, they are the bridge between the external pathogens as well as the internal immune cells. So the same thing has been incorporated, the same concept has been taken up. The only part is that we are only concerned with the binding part. We are not bothered with the effector part in immunohistochemistry. So another nice reason why antibodies are very easy and fun to work is you can actually isolate a specific antibodies. For example, intravenous, we have worked with intravenous immunoglobulins. Intravenous immunoglobulins are nothing but these are the pool of antibodies that we collect from normal human beings. And if you pass these antibodies through a Cephros column, having a particular antigen, you can actually identify, not only identify, but also enrich your material or antibody solution with a specific antibodies. For example, here we have made a oligomer A beta linked Cephros column, and when we passed through all the antibodies present in the human body, we could actually isolate the human A beta antibodies that are present circulating in normal human beings. So that was something very nice. So only the A beta antibodies can be easily enriched. And why is it? I mean, this sort of manipulation is also makes the antibodies very fun to work with, and also it becomes a great tool for the researchers. So again, there are 2 types of antibodies, as you all know. One is monoclonal antibodies and polyclonal antibodies. And to inform you, antibodies are the immunoglobulins that are produced by the B cells of our immune system, the B cells, B lymphocytes. Okay, so the property that's, and each B, the rule here is each B lymphocyte can produce only one type of antibody. Meaning each B lymphocyte will produce one type of antibody. That one type of antibody can recognize only one antigen. So if you have a collection of B lymphocytes, it will say 10 different B lymphocytes. These 10 different B lymphocytes can produce 10 different antibodies, and these 10 different antibodies can identify 10 different antigens. But then that's too broad. There is no specificity there. However, in case of monoclonal antibodies, this broad specificity has been narrowed down by isolating that single B cell, which is producing a single type of antibody that is capable of single antigen, and that particular antibody is taken up, and those B cells are fused with tumor cells. But again, normally, as I said, these antibodies are produced by the B lymphocytes, and if I want to produce some 5 liters of an antibody, I cannot draw 5 liters of blood from anyone. Okay, it's not ethical also. So what scientists have done is they fused these B cells with a tumor cell. Tumor cells can be grown in a dish, petri dish. So when you fuse a B cell with a tumor cell, what happens is that these B cells, they divide continuously. Remember, this is the same kind of B cell. So the same single B cell, when it fuses with the tumor cell, it produces antibodies, and all these antibodies will have a single specificity. So the specificity, these antibodies become very, very highly specific, and their binding affinities are very, very high. So that's the how the monoclonal antibodies are produced, and this is such a routinely used technique. For example, these antibodies produced, monoclonal antibodies, they are used as a, I mean, it has become a big industry in itself, right? And you can also patent, and there is a huge commercial value for these monoclonal antibodies. And not only that, even that's for research. Think about what happens if you develop an antibody that is going against coronavirus. That has also got a lot of huge amounts of commercial interest. So basically, the monoclonal antibody production is a very wonderful technique. It amplifies our ability to produce the same kind of antibody, the antibody with the same specificity, and to the extent what in the proportions we need. So that's about monoclonal antibodies. But then again, monoclonal antibodies not always useful. Sometimes we do require polyclonal antibodies. As I said, polyclonal antibodies are, these are the antibodies which are coming not from one single B lymphocyte, but a collection of B lymphocytes. Typically, polyclonal antibodies are produced from either rabbits, sheep, goats. What it is done, a small antigen is injected to the animal, and then again, after repeated exposure to the antigen, the immune system of this particular animal will produce antibodies, and these antibodies will be later on collected, and then again they will be enriched. So, however, this is little bit of the downside of polyclonal antibodies is that you have to have this blood collection part, which is not very ethical to do, and that's why we use goats, horses, donkeys, large animals, which can actually donate blood regularly. But then again, for that reason, monoclonal antibodies are better in the sense that you don't need any animal model, and it is highly non-invasive. So that's the place where monoclonal antibodies are good. But polyclonal antibodies are also their advantages. For example, if you don't know your infectious agent or your target properly, and if the antigen has not been characterized properly, then in that case, polyclonal antibodies are the best option to go. So even at the beginning of designing your immunohistochemistry, obviously you'll have to buy some antibodies. At that point of time, you have to really decide which antibody do you want. Do you want to go for polyclonal antibodies, or do you want to go for monoclonal antibodies? Remember, both have advantages and disadvantages. For example, just to give an example, if I have in a tissue, if I want to stain coronavirus, okay, if I want to use, do I want to use a polyclonal antibody, or do I want to use a monoclonal antibody? So if I want to stain only the spike protein of the coronavirus, only the spike protein, nothing else, then I would like to have a nice monoclonal antibody. However, if I don't have the crystal structure of the spike protein, and my antibody cannot avidly or faithfully identify only the spike protein, then it's always best to use polyclonal antibodies, wherein which the specificity for a mixture of antibodies. So some of the antibodies of the polyclonal antibodies will be detecting the spike, some other will be detecting envelope protein, some other proteins will be detecting some other portion of the coronavirus, but then again it will detect coronavirus. So here also if polyclonal antibodies detect the coronavirus, but then again the specificity is not restricted to the spike protein, but to other proteins present on the shell itself. So that's just an example to give you when to use a polyclonal antibody and when to use a monoclonal antibody. So whenever there is protein structure, you know what protein you are going to identify. Whenever its crystal structure is available, whenever a monoclonal antibody is commercially available, and whenever it is cheaper to use, just go for monoclonal antibody. But however, if monoclonal antibodies are typically they are very expensive. In such cases, if you are getting reasonably well results with polyclonal antibodies, with mild tweaking of your protocols, you can still get wonderful results with polyclonal antibodies. So this decision has to be made early on during designing an immunohistochemistry study. So this is actually a brief flow chart how this immunohistochemistry has to be carried out. So it has got three main parts. Okay, as you can see here, first is preparation of the tissue, then next is the actual immunohistochemistry part, and then finally the quantification. Because come on, you have stained the tissue, but nobody wants to have a qualitative information. I mean, if I stain my brain with A beta antibodies and I say, okay, there is A beta, okay, fine, that doesn't give much information. But then again, if I want to be quantitative, so you have to measure how much of the A beta protein is present inside the brain. That would be very, very useful. So in the same case, if I have an animal model for Alzheimer's disease, wherein which there is a deposition of A beta protein inside the brain, then not only I want to detect that A beta protein, I want to know where that protein is expressed, and also I want to know to what extent it is present. So for that purpose, you have this quantification, and with a caveat again, it's not an absolute quantification, it's only a semi-quantification. So you get a good idea, like 10% of my brain is covered with A beta protein, or 25% of the mouse brain, APPPS1 mouse brain is covered with A beta protein. So that kind of information is good enough for me. It already tells me that to what extent the pathology has set in. So just to walk you through some of the main steps of immunohistochemistry. So it all begins with the preparation of the tissue. With immunohistochemistry, if you sit down for 1 hour and plan it from starting to ending before priori, a priori means before you actually get down working, then it will be a lot easier. You will be able to troubleshoot a lot of issues that you might show up. I mean, issues can come from anywhere. It could be from the antibody, it could be from your buffer solution. So you have to sit down quietly and chart out the list of things, and then prepare notes or prepare how you're going to resolve it. So this is one of the critical things. If your tissue is not correct, if you are very careless in preparation of your tissue, then the next two steps, that is immunohistochemistry, is going to be very bad. The signal to noise ratio would be not at all satisfactory, and doing quantification in tissues where the signal to noise ratio is like nothing, then it would not give you a reliable result. So it would be waste of your time, it will be waste of your resources, it will be waste of the animal tissue as well. So that you don't want. So you better take care of this preparation of tissue in a proper way. Now I have seen when I came back from Sweden, in India I have seen that people are doing a wonderful job in terms of neuroscience and neuropharmacology. However, very few people actually perfuse the brain. So what they do is they open up the skull and they take the brain out, but then again they are not perfusing it. Now this is called as transcardial perfusion. This is very, very much necessary if you want to localize the CNS antigen. All right. The reason why it is because brain is highly vascular organ, and there are lots of immune cells. There are mouse itself antibodies are present, and if you are taking the tissue and if you are using it for homogenization or immunohistochemistry, it doesn't matter. If you don't get rid of the blood inside those blood vessels, they are going to obviously form artifacts. So, for example, you are measuring reactive oxygen species. You know that immune cells are rich in reactive oxygen species. And if your brain region is highly vascular, the readout that you're getting for those oxidative stress is actually coming from the blood, not from the brain itself, even though you have taken the brain region. So it is very, very important to carry out this transcardial perfusion. Transcardial perfusion is a technique wherein which you flush out all the blood from the within the blood vessels of the brain. So this is a small technique. I will show you how to do it. And then tissue isolation. Tissue needs to be isolated very quickly, very fast, because you know it's a tissue, it is going to degenerate. So you have to take care that it is always at +4 degrees. So not only that, any animal tissue or any protein for that matter in a biomedical lab needs to be at +4 degrees. No exceptions until unless it is fixed. Once the tissue has been fixed in formalin solution, it can sit on the table at room temperature. But if it is directly obtained freshly, it has to be stored at +4 degrees. So that so tissue handling is also very much important. Then the next part is the fixation part. How do you fix your tissue? Now the reason why you want to fix tissue is because it's a live tissue, right? And you have an antibody to detect it. So you want what this fixation does, it forms cross links between the proteins, as a result of which it enhances the antigenicity, or it enhances the antibody binding. So we'll see there are plenty of fixative agents are there. Not all fixative protocols are good. Some work, some don't work. We'll see which one works. And then also after fixation, there are also certain steps, right? For example, you have to in case of some tissues, you have to do cryoprotection, right? And those kind of things need to be taken care of. So if you take precautions on all these levels at the level of preparation of tissue, then you are good for going to immunohistochemistry. Next part. In the next part, the next part is actually immunohistochemistry. The main thing is the question you ask is how much of the antigen is present. For example, if I want to identify A beta protein inside the brain, the first question I should be asking is kitna A beta protein mere brain mein hai? Because that already tells you if the A beta protein is present in huge amounts, then probably I would like to do a specific, I would like to change the proportion of my detecting antibody. Right. However, if the target protein that I am trying to find in my brain is very minute, very small, and it is very unlikely that I will be able to find, then in that case I would like to increase the sensitivity of my workflow by using monoclonal antibodies, by increasing the concentration of the primary antibody. So this information about the amount of antigen is very much absolutely essential. Then again, there comes the techniques of antigen retrieval, how you bring out the antigen from the tissue. Then there are these blockage of endogenous peroxidases, as well as you have to block the non-specific binding of the antibodies. These are the techniques that we have to do so that the signal to noise ratio is enhanced. Now, what is signal to noise ratio in immunohistochemistry? At the end of the entire workflow, if you observe under the microscope the tissue, the target protein, the amount of signal that is coming from the target protein, and the amount of that constitutes the signal, and the amount of signal that is coming from everywhere else, that is the background, that conforms that is nothing but the noise. So the ratio of the signal to noise, that is very, very important, and you want the signal to be enhanced and the noise or the background visualization to be as low as possible, so that the signal to noise ratio is high enough so that the quantification would be much more easy. And for that purpose, you have to carry out the blocking of endogenous peroxidases. You also have to block the non-specific binding of the antibodies. Once you have done that, then you have to already decide how good you are at immunohistochemistry. Typically, I try to identify one antigen at a time. That's good enough for me. If I can identify one antigen, if I can identify A beta brain in the mouse tissue, that's good enough. However, sometimes I might be forced to identify more than one antigen. For example, I want to identify the A beta protein inside the brain, also the microglial protein also. I just wanted to see whether they're side by side to each other. Okay, so I might have 3 antigens at the same time. So at that point of time, I have to decide what should be the format of my immunohistochemistry protocol, whether it is single antigen or double antigen or multiple antigens, and how I should take care of it. And then format means, is it going to be direct immunohistochemistry, means the antibodies are directly tagged to the fluorescent molecules and I can actually visualize it, or it is not a direct one, it's an indirect one, meaning there is a detection antibody and then there is a what you call a tagged antibody. This tagged antibody detects the detection antibody and then it enhances or multiplies the signal. So those kinds of things are there that you have to decide which format you have to use so that you get an optimal signal to noise ratio. At the end of which, you also have to identify what are your detection antibodies, and also whether you want to use if, for example, you don't have a fluorescent microscope. Even if you don't have a fluorescent microscope, if you have just simply a light microscope, you can do beautiful immunohistochemistry images, and that you have to decide a priori before even starting it what you're using. If you don't have a fluorescent microscope, you can go for this DAB immunohistochemistry. That's wonderful, and you can also get in various variations. For example, DAB is a diaminobenzidine, and upon reaction, it produces a bluish brown product on the target protein, and this bluish brown product, it stands very, very out of the background, so it is easily visible. And this is sometimes DAP, as I said, is bluish brown, but then again you can make it red in color. Sometimes you can make it blue in color. So there are a lot of variations are there. So simultaneously you can use for a single stain or double stain as well. But fluorescence, if you want to do fluorescence staining, then it is you can do double staining very nicely. You can identify two different antigens simultaneously, and also you can look forward whether those two target proteins are present next to each other, or what is the distance between those kind of information can be obtained. So that's about the thing about immunohistochemistry. But then at the end of doing a successful immunohistochemistry, it's, as I said, it's incomplete if you don't do quantification. Quantification can be done by taking the images from using a microscope. Nowadays, when I did my PhD, I used to sit up to almost whole day behind a microscope and trying to get images. But these days, I have seen that you have these scanners are there. So all you do is you just feed your slide into the scanner. It will give you a high resolution image of the entire tissue and in say 5 minutes. So that's a welcome development, and I would love. I have used at Karolinska. I have used those scanners, and they're very wonderful, and they're better than I have taken images myself and also compared it with what scanner does. It's always better with the scanner because human intervention, wherever it is there, signal to noise ratio is not so good. There is a variation in that comes. But then again, in case of scanners, the same setting is ensures, and signal to noise ratio is always taken care of at the time of acquiring the image itself. So that's a very nice thing. And then at the end of quantification, what can you do for quantification? You can identify the profile that you want to identify in the tissue. It could be a cell, it could be a cytoplasmic cell, it could be a membrane protein, it could be simply blood vessels. It could be anything. It could be processes, it could be axons, it could be dendrites, it could be nuclei. So depending upon what profile you are measuring, you can either count them, or you can simply report as a % positive area. But then again, counting itself is a big area called as stereology. It will unbiased stereology. It itself will take more than 2 hours or a day together. It's itself a religion in neuroscience. So unbiased stereology. It's a wonderful science. If you have a time, just look at it. But then again, stereology for routinely we don't do it. But then if I really want to get a absolute quantification or a good estimate of a numbers, then in that case, the stereology is the one way to go. And then you can represent the data either as I said, % positive area, or simply you can report the number of profiles you can see in a given particular area. So along all the flow chart, all these different steps, you have to identify the sources for variation. Which source is most weakest one? Remember that place. If you correct it, you can improve the signal to noise ratio tremendously. So this is just a workflow. I'm not going to go through all the different steps. This is just a simple workflow. Now again, you really have to decide how you want to do your immunohistochemistry. This is a workflow for free floating sections, and then this is one for the paraffin sections. Now, which one to use? Do you want to use a free floating sections or a paraffin sections? Now, free floating sections is nothing but you section the brain, and the brain sections will be the antibody will have a chance to interact with the antigen present in the sections from all the sides, meaning the sections will be floating in the buffer, and the antibody will get a chance to identify its target protein from either side. So that's actually a very good thing. So I personally like free floating sections. Signal to noise ratio is wonderful with free floating sections, and also the ease of use is very nice with free floating sections. However, free floating sections doesn't work every time. For example, if I'm staining a protein which is present in very minute quantities, then probably I would like to go for paraffin sections, wherein which the paraffinized sections are already stuck to the slide, and you use this tray called as a staining tray, and the entire all the sequence steps of the staining are carried out on this staining tray. It's a staining chamber. Okay, so here the antibody will get to access to the tissue only from one side of the tissue. The other side is stuck to the slide. So basically, free floating sections are having an advantage, inherent advantage. But then again, paraffin sections are most widely used. I think very rarely here I think I have not seen anyone in India doing these free floating sections. Most of them, they prefer paraffin sections. One is it is very fast, but then and also it can identify if the antigen is present in very minute quantities. Probably it's advisable to go for paraffin sections. Not only, but then I don't like paraffin sections for one reason, because the antibody is going to get access from only one side. The amount of antibody that I have to drop inside for the staining is huge. I have to use say up to 1 to 100 or 1 to 200. The same antibody, if I use in free floating format, I can increase 10 times. I can save the primary antibody. I can go up to 1 to 10,000. So you see the difference. When the antibody has access from both the sides, you can save some antibody for yourself. Whereas, but then again, there it takes almost 1 day for staining. With paraffin section, you can stain it within 3 hours. So you save time in free floating sections, and also you save. You don't save time, but you save money. But here, in case of paraffin sections, you save time, but then you have to spend more money. So that's the thing that you have to decide which one is the best way to go. So this is what I was mentioning about the transcardial perfusion. We found that when you're identifying the antigens present inside in CNS, it's always better to perform this transcardial perfusion, because transcardial perfusion is nothing but this is a technique wherein which all the blood present inside the minute vasculature, brain vasculature, out with ice cold saline, with or without heparin. Now you add heparin because of that shock, those microclots might be formed inside the blood vessels. You can get rid of them. So with or without heparin, you can perfuse ice cold normal saline. Remember, you want to drive only the sterile normal saline, not the one you have prepared and you have stored it for 2 or 3 days and then use it. Because if the saline itself is having some particulate matter, they will end up in the tissue, and you will be able to see them in the immunochemistry, this thing that you don't want. So use a sterile ice cold normal saline. And how exactly you do the technique is, you just actually get access to the ventricle of the mouse. Okay, you make a small puncture in the ventricle of the mouse, you insert a needle. I mean, any 21G needle is fine, and then you can connect this needle to the ice cold normal saline. This is the one what has happened, and then you can puncture the right auricle. So whatever, and then you can switch on the perfusion pump. Perfusion pump is available there, you can buy it. They're available commercially, but then again, if you don't have enough funding for buying a perfusion machine, you can always use what you call as a gravity. For example, if this is the normal saline, you can put it on a certain height, and normal saline, the gravity itself will drive the normal saline into the cannula. So what happens is, the ice cold normal saline will be entering into this left ventricle, and via left ventricle it will be able to access the entire body. And then again, it will again rejoin here at the right auricle. Then you have to actually puncture the right auricle. So all the blood that is being circulated will be flushed out to the right auricle. So eventually, typically for a mouse, it takes around 3 to 5 minutes, wherein all of the blood inside the brain would be drained out of this right auricle, as a result of which is a good thing, but then again, it has to be faster enough, because if you prolong this transcardial ice cold perfusion, there is a chance that that itself might induce some artifacts. So that you have to optimize. So once transcardial perfusion is carried out, you can clearly see what would happen with or without transcardial perfusion. So this image, what you see is a mouse brain, and here this is what it looks like if you scoop out the brain without any perfusion. You can see all the blood vessels laden with blood, and they are present very much in the tissue. And if you take this tissue and homogenize it and measure, so you're not measuring what's inside the brain, but inside these blood vessels. So it's very, very much important to get rid of the get rid of the blood present in the brain vasculature. So this is towards your right is a brain that has been perfused. As you can see, once all the blood is lost or removed or washed out, it becomes a pale straw colored. I can still see a reddish tinges here towards the posterior surface, but then again, towards the caudal surface, there is still some reddish areas are there. But then again, this was done with gravity, meaning you're using the gravitational force. However, if you use a professional perfusion pump, you can obtain perfusion pump. What it does is it systematically pumps the normal saline just as its own heart will do. So there will be a pulsatile flow of the normal saline into the heart, so that the brain gets pulsatile, gets all this normal saline at the same pressure, at the same flow rate what this mouse heart pumps, and as a result of which perfusion will be nearly complete without any tinge of blood available. So this is very, very much important for immunohistochemistry, because if you have immunochemistry, if you cut those brain sections and your blood vessels are full of those antigens and they themselves have antibodies, then that's not a perfect way to carry out the immunohistochemistry. So it's very important to carry out a transcardial perfusion. I have a detailed protocol. Please let me know if you want. I can share it with you. So once you have carried out the transcardial perfusion, then you have the brain, and then the next thing is what do you do with that? The brain is there, it's fresh, and it's cold, obviously, because it has been perfused with ice cold saline. Some people like to, what they do is when they are perfusing it with saline, they also follow it with the perfusion with what you call 4% formaldehyde there itself. So the cannula also takes this fixing solution into the nooks and corners of the tissue, thereby optimal fixation can be done. You can do that, or normally what I do is I take this brain out and then I keep it in the fixation solution, which is nothing but 4% paraformaldehyde solution for 4 hours or overnight, whichever is convenient for you or not for you, for the target protein, and then you have to optimize whichever method is there. So immersion fixation is mean you take the brain out and you just simply immerse it in the 4% PFA. The one thing about PFA is that it's carcinogenic. You use gloves, use mask, and prepare it freshly from the powder. Okay, don't use PFA which is a day or 2 days old. Not good, because if there are particulate matter in it, then it's not good for immersion fixation. And prepare it freshly. The next step is also very much important. The next step is if you want to have a cryo sectioning, meaning if you want to cut the tissue at minus 20 in dry ice, or if you have not fixed it with 4% PFA, you directly want to get frozen sections, then before actually doing anything, you have to carry out this cryoprotection. Right? Cryoprotection means you are protecting the tissues from the ice crystals. So you all know that when a fresh tissue is taken and if it is dropped in a liquid nitrogen or a solid dry ice, what happens is all the fluid, all the fluid present inside those vasculature, it might actually crystallize into ice, and the same ice crystals, when you thaw them, they actually disintegrate the tissue. So the tissue, what you see here towards your right, is actually the ice, the freezing artifacts. This is a muscle tissue, and I mean, I have seen plenty of tissues of these kind of big vacuoles kind of thing. This means that adequate cryopreservation was not done. So the simple way to avoid this kind of a situation is simply once the tissue has been taken out, you have to immerse it in 30% sucrose overnight. So when 30% sucrose, it's a dense fluid, and since the water is less dense than 30% sucrose, as soon as you drop the brain into that 30% sucrose, it floats. So you have to let the sucrose get into the mouse brain, and as the sucrose gets inside the mouse brain, it settles down. And it settles down, and that means that the sucrose has imbibed into the brain, different brain structures, and that is good to go. So once sucrose is there, sucrose will avoid the formation of the ice crystals, and as a result of which, even if you're doing cryosectioning, or even if you are doing any cryosectioning, or if there is a thawing and freezing cycles are there, your brain will be adequately protected. So So you have to you have to decide whether you want to do it or not. I would like to do it because fixation is anyway you have to do it. So this is just another overnight of you drop it in the 30% sucrose, and then it takes up all the water present in the brain will be replaced by the 30% sucrose. It's sucrose, basic sucrose, nothing more than that. And at this point of time you have to make another decision whether you want to do a free floating section or you want to carry out the entire immunochemistry on the slide. So this way, actually, this is one critical area that you have to decide. I told you the advantage of free floating section, and the advantage of free floating immunochemistry is that you can save the antibodies, and the antibody penetration is much more better because it is penetrating from both the sides. Signal to noise ratio is great, but the disadvantages of free floating sections is that it takes time. It typically takes whole day. Or even sometimes 2 days or 3 days to stain. Whereas in case of on your right, the staining in a glass slide, the advantage is that it can be finished in a couple of hours, 4 hours at the most. And then but then disadvantage is antibody gets access only from one side. So signal to noise ratio may not be very good. But then again, another disadvantage of this one is that you need to dispense of huge quantities of antibodies. I hope I am audible. Everything is good. Hello.
Yes, sir. You are audible, sir.
Hello.
Yes, sir. You are audible, sir.
Professor Viresh.
Yes, sir. I'm here. You are audible, sir.
Hello.
Sir, you are audible.
Audible, huh? Okay. I just wanted to make sure if everything is okay or not. Okay. I will share my screen back again. All right. It looks like I'm audible. So let's move ahead. So we have already decided to do it whichever way we want to do, whether free floating or on slide. But then the next one is also we have to decide what type of sectioning you want to do. This is just a slide which should have showed you before. But then again, this is a typical sliding microtome. What you do is you fix the tissue here, and this is the blade. You simply slide it and you make thick sections. So this is the example of one is freezing cryostat, wherein which the inside temperatures will be around -20 to -30, and under those freezing conditions, you will section the brain tissue. So again, with for example, which you have to. I already told you what should be the thickness of the sections. Whether it should be 10 micrometers or less, or can it be between 35 to 80 micrometers. So typically what happens is thin sections are good for on slide, right? If you want to do your staining on slide, thin sections are good, and not 10 micrometer is too thick for it. Maybe 5 micrometers is an optimal compromise. Whereas, however, sometimes in most of the times in identifying or studying the antigens present inside the brain, you need to go for thick sections. I have used from 35 to 80 microns, and they work fine. And if I am using thick sections, I would like to go for free floating format, because the penetration of the antibody into this section, it takes time, and that time is available only with free floating format, not on the on tissue on slide staining. So again, whether I should the question you will be asking here is, what do you want? Either do you want a thin section or a thick section? Again, a part of answer is what format you are using, and also what is the profile that you are counting. For example, the cell that you are using, it is very big in size, and there is a significant what do you call contraction of the thickness, then probably I would go for thick sections. So that would give me a reasonably good compromise, even if during the clearing with silent treatments, if there is a contractions in the size of the or thickness of the section. So, and then comes the next step is the antigen retrieval. So previously we have seen that the tissue has been immersed in 4% formaldehyde. What this 4% formaldehyde does is it actually forms cross-links between the different proteins. Right. When there are different cross-links are present between the proteins, what happens is that your antibody, it is detecting an antigen. When an antibody is detecting an antigen, it is not only recognizing a specific peptide sequence, but also it is recognizing the conformation, the three-dimensional conformation, how different that protein molecule is arranged. So if it is already bound by the protein cross-links because of the formalin treatment or whatever might be the reason, the antibody will not be able to bind to its antigen. So in that case, what you can do is you can either heat the tissue section, or in the presence of sodium citrate solution at pH 6. For some strange reason, this is a universal reagent that can actually use for antigen retrieval. Nobody knows how and why. As soon as you treat these sections with sodium citrate and you heat it, I mean, for A-beta brain immunochemistry, I used to boil these brain sections in the sodium citrate at a pH 6 for about half an hour at about 80 degrees centigrade. So both the combined effect of heat at this particular pH 6, what happens is all these cross-links will be broken down, thereby the antigen will be retrieved back, and the antibody or your detection antibody would be able to easily locate the antigen. So antigen retrieval step is very much important. It enhances the signal to noise ratio. You always want to enhance this signal to noise ratio. So the next part is the tissue is a very rich source of endogenous peroxidase. Peroxidase is an enzyme. For example, if this is the antigen that you want to identify, and there is a lots of peroxidases, peroxidase is also unfortunately the same enzyme that is also present here in the detection system. As a result of which, there is a very likelihood that the peroxidase of the detection system and the peroxidase are present on the tissue, they might actually give you a false positive, as a result of which signal to noise ratio will be going down. So it is very much important to block this endogenous peroxidase activity, and it's very easy to block this endogenous peroxidase activity. Just treat the sections with 3% H2O2 for half an hour in a free floating format. It should block all the endogenous peroxide. You will get a beautiful immunohistochemistry if you have blocked with this H2O2 treatment. So that is something we have seen consistently, and I advise always to block the endogenous peroxidase activity if your detection system is having a peroxidase component here. So that's also one step where you can actually increase the signal to noise ratio. The other part is, as I said, that you also have to block non-specific binding. Now, what is non-specific binding? In immunohistochemistry, you're using antibodies to detect the antigen, and which you think that it is very specific. Very true. The FAB portion of the antibody is capable of detecting it, but then again, unfortunately, the antibody also has this FC portion, the tail portion, which not only it actually has got receptor. Every tissue has a receptors for this tail portion. This is called as FC receptors, and these FC receptors can bind to your detection antibody. As a result of which, your detection antibody is not only detecting the antigen, but also the FAB. So your detection antibody has got an FAB portion. This FAB portion of your detecting antibody is not only detecting the antigen of your interest, but also the FC portion is also interacting with the tissue FC receptors. As a result of which, you will get a signal from this antigen. Now also you will get a signal from a background also, which you don't want. So this is again gives a false positive, and again it brings down the signal to noise ratio. So you have to have a very way to get rid of this interaction. It's also very easy. It's not very difficult. What you can do is these are very low intensity. I mean, these are weak interactions, and you can easily block them by using 5% normal growth serum or donkey serum, or sometimes people have also with normal bovine serum, albumin also. So what these normal goat serum does is, these antibodies, they will not bind to the antigen, but then definitely they will bind to or cap all the FC receptors present in the tissue. So when you actually drop in your detection antibody, it has got only the antigen specific sites, but all the other sites have already been blocked by the non-specific, blocked by the normal goat serum. So it's advisable that once you have used normal goat serum, not to wash off, because immunohistochemistry, after every step you have to wash continuously in the washing buffer. So once you have this normal goat serum, don't wash it off, because once you're washing it off, then basically you're getting rid of the FC, getting rid of this interaction between the FC receptors and the FC portion of normal goat serum, which you want them to be there. So this is another way how you can create the signal to noise ratio, and this is also a very important part in immunohistochemistry output. Now then comes which antibody shall I be using, and this is the most critical part. So sometimes there are plenty of antibodies available in the market, and literally every company has a primary list of primary antibodies and list of secondary antibodies, and then you always have the question which one to use. So the choice is simple. So if you're using a mouse tissue, you don't want to use a primary antibody that is produced in mouse. No. So the rule of thumb, of the golden rule says the species that are the species of the target tissue, the species of the primary antibody should be far away from each other. Meaning, for a mouse tissue, I don't want to use a mouse primary antibody. I would be happy with either with a rabbit antibody or a donkey antibody. In the same way, if I have used a primary antibody which is a mouse tissue or a rabbit tissue, I don't want to use a secondary antibody which is a mouse or a rat. So as I said, the species should be as far as possible. So here, if the primary antibody is a mouse, maybe the secondary antibody would like to have a donkey or a chicken for that purpose. For example, if I've used a primary antibody as a rabbit, I don't want to use a secondary antibody as a rabbit at all. I would like to have a mouse. So these species should be far away from each other. Okay, the species difference between the primary antibody and secondary antibody should be wide enough. The species difference between the primary antibody and the target tissue also should be wide enough. Then only it can result in good signal to noise ratio, and also it will avoid all sorts of non-specific bindings. So this is again a decision point you have to make. All these commercial entities have got a wonderful what you call wonderful mechanisms or wonderful flow charts that can guide you which antibody to use, whether you should use a mouse monoclonal or a rat monoclonal or a donkey polyclonal or a goat polyclonal. I already told you how you decide whether you want to go for a monoclonal or a polyclonal antibody. Again, the cost is another important factor. Monoclonal antibodies, they cost something around primary antibodies, and speaking of which, they may cost around 300 euros. But then that's quite a lot. But then again, these secondary antibodies also try to buy only from one specific source, test them. They should not have any background staining, and then only you buy them. And then again, they can cost around $150 each one. But then again, if you use in a free floating format, you can save these antibodies. So next, then you have to decide which IHC format you have to use. So there are different formats are available. So this is just a simplification of what are the different types of formats are there. The first one is called as a direct method. In direct method, it's very simple. There is an antigen present on the tissue. There is an antibody to detect this antigen, and this antibody itself is bound to an enzyme or a tag. So basically, it's a one-step process, and you simply drop in the antibody and let it bind to this antigen, and then you're good to develop the color. So this is a direct method. Direct method, as you can see, every single antigen will have a one single molecule of an enzyme, or that might give you 1 unit of signal. But then again, that's not very good enough if the antigen is present in very small amounts. It's okay if antigen is present in huge amounts. So typically, normally, we don't use direct method because its sensitivity is not very good, and also we don't want that. Because whenever the primary antibody is labeled, its ability to detect the antigen also invariably gets affected. So most of the times we go for indirect method, wherein which we leave aside the primary antibody. For example, in this indirect method, there is an antigen here again, and then there is a primary antibody, and this primary antibody binds to this antigen, and then the secondary antibody will bind to the primary antibody, but this secondary antibody has this enzyme. As a result of which, you can see a single antibody can actually bound to two different enzyme secondary antibodies, and these two different secondary antibodies might result in 2 units of signal because of the visualization. So basically here you can see the signal amplification twice than that of a direct method. But then again, this is also good enough. Sometimes you also have what is called the triple step IHC method. So as you see, you are increasing the sensitivity. So initially we saw what with the single antigen molecule, if there is a single unit of signal, now in this 3-step with a single unit of this antigen, there could be a multiple units of signals. So this is a triple step wherein which there is an antigen, there is a primary antibody that binds to these antigens, and then there is a secondary antibody that is biotinylated. This is the biotinylated secondary antibody, and then bound to this biotinylated secondary antibody is this streptavidin enzyme complex. These squares are streptavidin enzyme complex, which actually has multiple enzyme molecules. So whenever for every single unit of antigen, you can see how much signal it is giving. So it is actually amplifying the signal, which is very much desirable. So typically in a routine, try to use this 3-step IHC method. It enhances the signal to noise ratio, and just to. And then again, you also have this comfort of using double staining. For example, if I want to stain both the amyloid plaques inside the brain and also the neurons, then probably I will have to go for double staining. In case of double staining, there are 2 different antigens. This is the first antigen. This is the circles and the triangles. So what you do is either you can detect them sequentially or simultaneous. It's up to you. So the format that is shown here is sequential format, wherein which the first antibody is bound, and then it has a signal has been generated because of it, and then a second antibody is primary antibody detects these circles, and then it produces its own signal, and then thereby both the target proteins can be studied, can be evaluated, can be visualized simultaneously. So this is the format that you have to obtain if you want to do double staining with immunohistochemistry. It results in beautiful stainings if you go for fluorescent ones. So this is just a brief summary of what are the different formats of immunohistochemistry that are available. So there is a direct method, as I said, and there is an indirect method, and then there is the 3-step methods are present. And then latest, or I have this one I have used recently, wherein which the secondary antibody here it is bound only to the one molecule of enzyme. Nowadays, thanks to the polymer technology, the secondary antibody is attached to a big polymer containing hundreds of these enzyme molecules, as a result of which every single unit of antigen can result in multiple signal units. So signal will be amplified, will be enhanced several hundreds of times. That's a beautiful thing to do. Because why? Because sometimes the antigens are very scanty, very little. Sometimes they're very good, they're very huge. In that cases, you don't need a simple signal amplification. This kind of method would be suffice. But however, if you want to identify a protein which is not, for example, you are looking for neurogenesis, the birth of newborn neurons, they are very less in number, and double cotton is the target that you are detecting. And if you want to detect it, I would advise go for this polymer based signal amplification method. This would give you that each and every double cotton molecule present, the signal will be amplified several hundred times. So this is just you have to choose the best format. It all depends upon trial and error. You have to always run some pilot staining this thing, and then all you see is signal to noise. What is my signal and what is the noise? The signal should be more, the noise should be less. And also, as I say, it involves several steps. There should be ease of handling. Right, it should not be undue stressful for the person who is doing, and also you have to think about the costs of the antibodies, the costs of the reagents. So it is quite a compromise. But then again, remember always you will enjoy immunohistochemistry only if you make it enjoyable. Of course, and if once it is enjoyable, you're ready to take up whatever challenges it throws at you. So that's about the IHC format that you have. And then another important part for not just immunohistochemistry, any detection system is controls. Controls is very, very important. Why? Because what you're seeing is nothing but what your primary antibody is seeing, right? And what your primary antibody is seeing, you want to make sure that it is seeing what it is supposed to see. Right? If I'm using an antibody for an A beta, I expect that antibody to detect only A beta. I don't want it to expect it to bind to APP protein, right? Which also has A beta sequence. So I should have proper controls. And trust me, your reviewers will be very harsh on you if you don't employ sufficient controls. For example, you should have a positive control. For example, if you are detecting a human A beta protein, there should be, in fact, as one slide showing that there is a beta protein and this antibody is capable of detecting it. So that's the positive control. And then you have a series of negative controls. There has to be a negative control for the procedure itself, meaning in the entire workflow, you will avoid primary antibody, but all the sequence will be going on the same. So as a result of which, that should be a negative control. At the same time, if you have access to a knockout mouse wherein which the target protein itself is absent, so then in that case, that should be a negative control. So that should also, you should not see any signal from that. And then again, it's also better to have a control for the detection methods, because you really want to know where the signal is coming. For example, this is true especially in case of a fluorescent tags, if you're using fluorescent antibodies. So I mean this fluorescent antibody, once they have been tagged, their specificity changes and it starts binding to non-specific regions also. At that point of time, it is better to have this kind of get rid of this control also, that tells you to what extent the signal you're seeing is contributed by the noise. So IHC controls is all about the noise. Noise should be minimal, the signal should be maximal. That should be the mantra here. So this is a quick summary of what I have shown. We first perfuse the animal, get rid of all the blood from the brain, and then you take the tissue and you can process the tissue accordingly. Either you fix it in formaldehyde, or you can freeze it, whatever you want to use. But then again, if you want to freeze it, better do cryopreservation. And then once it is there, if you would decide to go for slides, then get them onto a gelatinous slide, stick the tissue, then go for this antigen retrieval, then block with either the normal goat serum or BSA, and then start your staining procedure. Use your primary antibody, and then again, to detect this primary antibody, either you can go for DAB. It depends upon which microscope you have. If you have a fluorescent microscope, use a fluorescent antibody tag. If you don't have a fluorescent microscope, go for chromogen detection using DAB staining, and as a result of which, at the end, you will find the staining. And then here, this is all the black here is the background, and all that red, what you see is the signal. The signal to noise ratio should be clear. And in the case of DAB, all the signal is shown in bluish brownish in color, and all the background should be perfect white in color. So this concludes what you call is the workflow for the immunochemistry. But then again, as I said, it is incomplete if you don't do image analysis. So you want to really quantify it. You, at the end of the entire process, you want to give some numbers. You want to throw some numbers and say, okay, because of these numbers, there is too much of a beta in the brain. You have to give those numbers. Those numbers can be obtained by using image analysis. But then image analysis is a very tricky situation. It itself will take another 1 hour. But then again, we don't have initially during when I discussed with Viresh sir, I thought that if you all have access to Photoshop CS3, I can actually show you how to do this. But then again, I have just borrowed few slides for now. But then again, image analysis is something where your integrity will be questioned, and it will be checked very seriously. So while manipulating the images, all the changes are permitted that are global. Meaning you cannot specify or enhance a particular portion of the image while suppress another portion. That's not acceptable. If you want to change an image, do a global change, right? And then local change of adding, removing it is not advisable to do. And then all the whatever image processing steps you do, it has to be documented. And most of the times it is, do we have to tell them or do we have to not? That ambiguity is there. And whenever in such a confusion arises, the best thing is to tell them whatever you have done in the form of a protocol. So these are the things that you have to always keep in mind. And yeah, this is the place lots of the complaints are coming from. People are using dubious methods to show what they want to see, because reviewers would like to see the results, and it's with image analysis, it's very easy to manipulate it. So you shouldn't deviate from your focus here. Okay, integrity should be safeguarded, which is the paramount. Now I would quickly show you how you can do. This is an image of a blood vessel that was taken from the brain. You see the signal to noise ratio is good. One rule of thumb is whenever you are using here I have used Adobe Photoshop, but then you can do all these things even in ImageJ. ImageJ is an open source software. Okay, so whenever the basic thing that you can do for image processing is you can visualize the histogram of the image, and you try to make sure that the entire histogram is from 0 to 255. As you can see, these pixels are here missing. So what you can do is you can simply stretch this bar and make this yeah. So I will show you how to make an ideal image. So what you can do is, once you have on Photoshop, you can do it whatever after you have taken the image, right? Or you take the image on a microscope with a camera fitted to it. But then again, if it's not ideal one, you can change it on Photoshop later on. But the best way to do it is change the image already when you are on the microscope with the camera. So this is the camera that I have used. This is software I have used. So this is the histogram. As you can see that it is not stretched appropriately. So I have to, these are the shadows, these are the zeros, and these are the 256. So I have brought it down a little bit so that white should be white, black should be black. This is the golden rule. Whenever you're adjusting the histogram of an image, white should be white, black should be black. So everything towards the left is blacks and everything towards the right is whites. And you can play on with the sliders. Remember, whenever you're sliding these using these tabs, you're making changes to the entire image. You're not specifically doing manipulating a single portion. So that's you can do already at the time of acquisition. That's always advisable. So this is if your camera is not good for some reason, you can also do it on Photoshop. In Photoshop, there is an option called as the levels, wherein which here you see for this particular image, these portion of the pixels are missing in the histogram. The histogram starts somewhere here. So what you can do is you can simply slide it so that your entire 0 to 255 is spanned by the same image. So you can see already the difference between the signal to noise ratio, it is already enhanced. So this is how you can make a global correction for the image. There is another wonderful feature with Adobe Photoshop C is, for example, in immunochemistry, you have, for example, if I don't have fluorescent microscope, I have a normal light microscope fitted with an old camera. But then again, at that point of time, it would be very difficult for me to identify a particular profile. For example, this is have shown, this is different nuts, and they are all of different colors. So I just took a pic of it. One important feature of Adobe Photoshop is that whenever you apply grayscale, what it does is it treats all the colors in the same way, right? So grayscale means everything is in grayscale. But then again, in Photoshop, there is an option called the black and white, wherein which you can actually decide which of these colors needs to be darker and which of these colors needs to be lighter. So typically in a normal grayscale mode, what you see is that the contrast here with the cover is very less, whereas the contrast is very, very high. So whenever you're working with, obviously it is easy to work with grayscale pictures, but then again, whenever you're making grayscale, you have the option, extra option with the with Photoshop CS3, where you can actually play with which color you want it to be darker, which color you want it to be lighter. So this grayscale black and white option is a wonderful one if someone wants to try. But then again, remember, the entire image will be treated in the same way, not just specific to this particular nuts. Okay. Another useful feature of this grayscale using this black and white is, this is a brain section wherein which we were trying to see if there were bleeds inside the brain. So all those bleeds would be appearing in the form of these hemosiderin deposits, which are dark blue in color. And since this, when I normally do a typical grayscale, what happens is it takes the same color as the background. So even the nuclear also gray, and even the blue color is also becomes the gray, same gray. It becomes very difficult to differentiate between the background and the signal. So signal to noise ratio is less. What I can do is, go to the black and white option, I can make it to lighter, the blue portion much more lighter, as a result of which all the blues here becomes conspicuous. Then I can pick up this color for quantification and say, okay, this much area of the brain is having blood clots. So this really gives a powerful tool to isolate the regions of interest and bring out the pixel information, and then you can easily inform it. So another nice feature about Photoshop CS3 is that you can actually do stitching. Previously, I remember in my PhD when I was doing, I used to sit for months together under, behind a microscope, trying to take images of the whole mouse brain. So, but then if I want to get a higher resolution, obviously I won't be able to capture the entire brain in one go. So I used to do section 1, section 2, section 3, section 4, and then used to analyze individually. However, Photoshop CS3 has got this photo merge, wherein which you, after taking it, one single click of a button, it will stitch all the photos together. I think your iPhone and even Samsung phones have this ability, the photo merge option. So now in one go, I can measure the entire plaque load inside the brain. This is the cortical region. In one go, I can report it out. So this is one other very important or a nice feature that I like very much. But if you really want to separate out the different colors on the section and analyze them, there is another option called as actions in Photoshop. For example, I used to have some close to 400 images to analyze, right, from single project. And remember, if each of which takes me 10 minutes, it will take forever for me to do all those things. However, with actions in Photoshop, repetitive steps can be automated. So as a result of which, these set up for 20 minutes, 20 steps, it used to take somewhere around 15 minutes. But then because of these actions, I can do it in 2 minutes easily. So it saves a lot of time. So that's also one of the powerful feature available with Photoshop CS3. I believe it is. These options are also available in ImageJ as well. Whenever in future we get an option, I will show you hands-on how to do this. So I have shown you everything about the immunochemistry. Now I just quickly want to show how I have used it. So this is one slide that shows there was a question by my reviewers whether the antibody that I was treating the mouse with, did it really enter the brain or not. The question here was whether the antibody that I was giving a human antibody to a mouse, whether it actually really got released into the brain or not. So what I found was I used an anti-human IgG immunostaining in the mouse brain. This is a control section. As you can see, there is no staining whatsoever. However, here this is the hippocampus, and there is an accumulation of the immunostaining from the rostral side towards the caudal side. So there is an accumulation of IgG inside the hippocampus of the mouse brain, and this I can prove along with showing the negative control here. But then again, our reviewers were not convinced. So what we did was we did a wonderful triple staining. So what we did was we detected the human IgG with a green fluorophore, and then we also detected the A beta deposits inside this mouse brain with a red fluorophore, and then we also wanted to see whether it is surrounded by microglia, and hence when you merge them together, you find that human IgG which is present within the same confines of microglia and A beta local area. So this gives you a mechanism that human IgG or antibodies which are given to the mouse, they were capable of detecting the A beta present in the mouse brain, and then because of which microglial cells have actually been activated. So there is a binding function as well as there is an effector function. So it has really given so many insights into mechanistic insights. So immunochemistry has really helped us pacify that reviewer. So this is another one I have used. This is an immunochemistry. You can see the signal to noise ratio. The signal is all those dark spots or the A beta plaques present in the cortex, and this is birch wing shaped hippocampus, and you see that there is a beautiful signal to noise ratio. And in this particular slide, we followed these mice for 12 months and see that there is a progressive deposition of these amyloid plaques. And the question we asked was whether the having or not having a P50 gene, whether it had any impact on the amyloid plaques, and as you can see that there was no impact of this P50 gene. So basically immunohistochemistry has actually answered whether the P50 gene takes part or participates in deposition of amyloid or not. The answer is clear no here. So that's again thanks to immunohistochemistry. Immunohistochemistry not only about the amyloid plaques or the neurons, it can be also used in thick sections. So these are the sections, sagittal sections from a mouse brain, and these were actually scanned, not taken from the microscope. The entire brain from the rostral side to the caudal side, they were scanned, and what you can see clearly a very nice signal to noise ratio. So these are the microglia EBA antibody stained ones, and you can see these are the pacified microglia quiet ones. Here also you can see next within a 500 micrometer, you can also see a microglial cell which is very dark and rigorous. right? So these are this tells that my staining is working very nicely, that it can differentiate between two microglial cells, one expressing very low amounts of EBA and the other expressing very high amounts of EBA. That means that signal to noise ratio is very, very nice. So this is another high magnification images of the same, just for you to appreciate the the cellular structure of these microglial cells. The microglial cells are known to be very dynamic. They change their shapes in a jiffy. They could be quiet, or within a second they could be a very activated ones. So this is just this is from hippocampus, this is from substantia nigra, this is from lateral geniculate nucleus region of the brain, and as you can see, the same EBA1A protein present in the microglia is differentially expressed in the microglia. That also tells a lot about its biology. So this is from striatum, if I remember right. These microglia had actually formed trains, and these are actually the white matter tracts. So microglia are actually enveloping the white matter tracts, which is a wonderful thing. I don't know the biological consequence of it, but then it already tells that microglial cells have a role in transmission of the nerve impulses, or preserving the or maintenance of insulation. But then again, there is evidence that too much of microglial activation in and around the axons is also not advisable. But then again, these are from the normal mice, and let's show you other. Yes, this is again a microgliosis, and this is the choroid plexus here. And this is an example of a double staining here. Here I have stained the amyloid plaques with Congo red, and then again I have stained the microglial cells with EBA on the same. And as you see, this was stained with a DAB chromogen, and this was a Congo red histology stain, which stains it the plaques as red. As you can see, the same information what you could get with a fluorescent microscope, you can also get it with these with a normal simple microscope, light microscope. This is again this is the region. This is something I'm very proud of. I was able to convince the reviewer that the cells, the round cells that I see inside the brain, they are actually the infiltrating cells coming from the periphery. We have used CD45 as a primary antibody, and you can see that these CD45 cells are actually exudating, entering into the brain parenchyma via these blood vessels. And then also you can also see the CD45 positive cells on the meninges, and in the parameter also they are also entering via this route. And this is again a double stain spider-like microglia. Microglia, as I said, they have different phenotypes. They could be spider-like, or they could also be round shaped. They're all coming from their actually monocytes in the periphery. They transform themselves into microglial cells. So this is the one staining that we have done in Stockholm at Karolinska. This is the tissue from human postmortem brain. So towards your left side, this is the brain from a person suffering from Alzheimer's disease, and towards your right is the brain of a person, postmortem brain of a person suffering is a normal control. So he's not suffering from any, he was not suffering from any Alzheimer's, but then again, he died of some other reasons. But then again, clearly you can see there is a day and night difference between the person suffering from sporadic Alzheimer's disease and a person suffering from, not suffering from, and it's controlled. So these are the different primary antibodies that we have used. The first two, 82E1 and EBX42, as you can see across the 6 layers of the neocortex, you can see large amyloid plaques are distributed all along the neocortex and along all the layers. The same in the case of a normal person, they are not present. These people, they have memory issues, they have dementia, and these guys, they don't have any cognitive deficits. But then again, even in normal cases also, you find the amyloid plaques, indicating that amyloid plaques, after all, they may not be the targets here. However, the biggest day and night difference is seen with tau staining, 88, CP13, PHF1, MC1. These are the primary antibodies that can be used to stain phosphorylated tau protein, which is also present inside the neurons. And as a result of it, you can see the neurofibrillary tangles, these flame-shaped structures. As you can see, they are present across the different cell layers, more likely in the layer 3 and layer 5, layer 4, I think, layer 5, I think. Yeah, so, but then again, in case of controls, there are absolutely no tau staining. So basically, it tells you that tau, the formation of neurofibrillary tangles, it is way more critical in assessing the Alzheimer's neuropathology rather than just merely using amyloid-based neuropathological techniques. So that was about the postmortem human brains. I think I have given you I have given you the examples from mouse brain. I have given you examples from different antibodies. I have given you examples from human brain. I have walked you through the different steps of immunohistochemistry, and here are some of the resources what you could find from the internet. And yeah, I think thank you very much for your patient listening. I hope if there are any questions, I'll be very happy to answer. And here is my email ID if you want to any point of time. If you need any advice or if you need any help with your protocols, I'll be more than happy to contribute. Thank you very much. Sir, I think I have concluded my presentation. I will stop my presentation, and if there are any questions, I will be happy to take some questions. Over to you, ma'am.
Thank you, sir. Thanks a lot. Your presentation was very good, very informative, in-depth, very impressive presentation, sir.
I hope.
Thanks a lot. Thanks a lot. And before we go to the question and answer session, I would like to invite our principal and convener of this program, Dr. Madhav Reddy, to give his note to the participants. Welcome, sir.
I think there is some problem with the voice.
Yeah. Looks like there's a technical issue again. We'll just check with it, sir.
Yeah.
Over to Jayanti, madam.
Sir, looks like there's a technical issue. Again, we are facing the same technical issue. We'll come back to sir's speech later. We'll continue with the question and answer session. Over to Dr. Padmavati to continue the question and answer session. Welcome, Padmavati.
Thank you, madam. Hello, Dr. Lakshman sir. Naresh? Thank you, sir. It was really simple and outstanding presentation. And so we have few questions from the participants. They want to clarify some doubts. And before that, they have given many compliments, sir. So your presentation was nice, informative, interesting, very helpful, very prospective, and good work. They're very much impressed with your presentation, sir.
Thank you, ma'am.
Shall we proceed with the questions?
Sure. I'll try my best to answer them.
Naresh? Can you see the questions on the screen?
No, not yet. I just see the home screen yet.
No, not yet, sir. Because I'm asking Naresh, sir, that
okay.
are we able to see the questions or not? Sir, one question is there from J. Bhaskar. Sir, monoclonal antibody technology, how it is useful to reduce the side effects of cancer therapy?
Well,
yes.
Actually, the main focus of this presentation was using antibodies as a research reagent, right? So for detecting the antigens on a tissue. But then again, in cancer therapy, if there is a monoclonal antibody, if you really want to know whether this monoclonal antibody it is actually capable of decorating or if it is capable of binding to its target region, then obviously you can design a simple immunohistochemistry using the same antibody that was used for therapy, and then again using an appropriate secondary antibody. And then one thing that you can prove using this tool is that whether the therapeutic antibody can bind to its target or not, and to what extent it can bind, and what are the different tissue structures. It can also tell you whether your therapeutic antibody is binding to its target antibody, not only target antibody, whether it is capable of binding other structures also in the body. For example, if an antibody is capable of binding cancer cells in the breast tissue, so is this antibody is also binding somewhere else, also in the brain, also in the kidneys, also, which you don't want. So that could also be checked using these immunohistochemistry tools. And I think for diagnostic purposes, it is very routinely used, and the quality is should all the slides that I showed you, they are from academic perspective, meaning the quality is not to the, but then when you're using it for diagnosis, the control should be really perfect, 100% sure. So it would greatly help in drug discovery when it comes to the detection of cancer cells using a therapeutic antibodies. If I hope I've answered the question.
Thank you, sir. So next question we have from Lingesh Allakonda. So what are the advantages and limitations of IHC in comparison to Western blotting? Which one is better for research activities?
See, Western blotting also, the basic concept is same. The antibody is detecting the antigen. But then in Western blotting, what you do is you take the tissue, you mash up the tissue, homogenize it, and you try to blot it, right? Either on the gel or on the paper, nitrocellulose paper. But then again, there you get only the answer is whether the protein is present or not, yes or no. But in case of immunohistology, in addition to the yes or no, as I said, it can also tell you where that protein is present, whether the protein is present in cell A or cell B, how far the cell A is different from cell B, in what form it is there. So all this, and to what extent it is there. If there is a high intensity of staining, it might already tell you that the expression of that particular protein is high. So it gives you the cellular context, it gives you the micro, it gives you information about the micro tissue microenvironment of the target protein, which is very much essential for understanding the disease biology or therapeutics, whatsoever you may call it. So yeah, it is, I mean, typically immunohistology before actually Western blotting, I will prefer immunohistology for than just simple Western blotting, because Western blotting, even though it's in quantity, doesn't give you the contextual information. The contextual information is given by the immunohistology. So I would go for immunohistology only.
Thank you, sir. So next question is from Ruksar Fatima. How much do cost an IHC study for detecting a single antigen, as it involves a very sensitive procedure to follow?
Yeah, that's a very not a simple question. It's a very difficult question, because it all, see, let's be frank. The mainly the reagents that are going to cost expensive are the primary antibodies, secondary antibodies, and the detection reagents. See, microscopes and all these you can borrow the facility, but then again, these reagents that are the primary antibodies and secondary antibodies and the detection reagents, that you need to buy. And typically, any primary antibody, again, if it is very widely used, you might get it for, say, I don't know in India, but then in for a decent 300 euros you can get a good antibody, monoclonal antibody. But then again, polyclonal antibodies are relatively less expensive than monoclonal antibodies. If it is available, you can go for it. But then again, also try to, if you spend some money on these secondary antibodies, these antibodies can be used for a long period. I mean, what I used to do is, if I used to spend 300 euros on a primary or a secondary antibody, I used to aliquot them in 10 microliters, keep them in -80. Whenever I want, I used to use them. Sometimes, if you are having a staining the entire week, you can actually preserve that antibody solution. You can reuse it, especially for free floating. So, depending upon your resources and the amount of money you can spend, you can decide whether you have to spend more time or less time. So, yeah, it is. I agree with you, but then again, when compared with other But then again, these are the basic things. That is primary antibody, secondary antibody, reagents. Microscopy, even with a light microscopy, also you can do wonderful things. And ImageJ is an open source platform, so you can easily get numbers. Yeah, with the 50,000 rupees, I think you should be able to buy the detection reagents and secondary antibodies. But then primary antibodies, I cannot tell the price because it depends upon, for example, one EBA antibody that I showed you in the slide, that single 100 microliter, it costs around 600 euros. And also, if you look at the A-beta antibody that I've shown, that costs only 150 euros. So it depends upon the primary antibody. And also, sometimes, most of the times, what happens if you write directly to the person who has produced this antibody, he might actually send you a small sample of it. It happened so many times that I was writing to the authors, and they used to spare a small amount of antibody, which used to use it in our lab. So yeah, it all depends upon your willingness to take chances.
Thank you, sir. Next question is from Meghana Sweetie. Whether flow cytometry and IHC data comparable? If yes, what are the parameters to be considered?
Flow cytometry, again, flow cytometry is a very widely used tool, and especially for immunology. My experience hasn't been so good with flow cytometry, especially with this when I was dealing with these microglial cells or immune cells, because the extraction of the immune cells, the initial tissue extraction itself, is so stressful that it pushes those cells into activation forms. And I don't want to do anything to the tissue which actually activates them, especially when I'm dealing with immune cells, which are very dynamic in terms of their activation profiles. So I did not like it. But then again, with flow cytometry, you do not get the contextual information. For example, a sample has got a high amount of CD45. You really don't know whether the CD45 signal is coming from within the brain or within the blood vessels, right? You really don't know whether that CD45 is coming from a cell that looks like a microglial cell, or from a cell that looks like a round cell. So the contextual information is missing from flow cytometry. So that's the main difference between flow cytometry and immunochemistry.
Thank you, sir. Next question is from Lalita Devi. Is there any possibility of co-stain in IHC technique, and suggest the precautions to avoid such co-stains?
Co-staining means double staining. If she means double staining, yes, you can do double staining, meaning you're trying to identify two different antigens together. But then again, I think I have shown one slide. You can either use primary antibodies sequentially, or you can use one after the other. So I would personally do it one after the other. I will first complete one staining, and then I will after being happy with the quality, I will proceed with the second steps of staining, and I would do free floating format. And always you have to see whether, if you're using fluorescent tags, try to use contrasting colors, the red with the green and the blue. And if you're using DAB based chromogens, try to differentiate between the red, blue and brown. So that is possible. With the addition of the nickel in the DAB solution, you can make the final product more dark blue. So that would be quite in contrast with the just brown DAB color. So playing with them, you can actually really find this, carry out this co-stainings. And as I shown you, I have combined it with the histology, congruent histology. So it's perfectly doable. But then again, you have to play around, you have to enjoy what you're doing. There will be failures, but then again, you will succeed at the end.
Thank you, sir. I think next it is not question, is trying to give some information. Sir, IHC detection is available in commercial lab in India in field of CVS toxicity study. I don't understand.
Yeah, so what Dr. Kushwa Ajay Singh is trying to say is that, yeah, basically immunochemistry, all diagnostic labs do it. So if you tell them, give it to any diagnostic lab, but then again, any contract research organizations also should be able to do it. But then why you want to give it and pay someone when you can do it here? Where is the fun? The beauty of being a researcher is that you try to learn in your own lab. You try to learn about things, but then again, without any commercial angle. So if I'm not motivated by money, then I'm more prone to look around things, play around with, and maybe develop a new one or see something new information. Yeah.
I think there are no more questions, sir.
Okay.
Let us see whether the principal sir is available or not once again.
Oh, all right, all right.
Naresh? Naresh, sir? Yes, sir. Now he's in live. Sir, is it audible? Principal, sir. Sir, are you able to hear me, sir?
Madam, there may be a technical problem. It seems. You can continue.
Okay, sir. Okay, sir. Sir is
No,
we are not able to.
Yeah, it happens. I mean,
Yes,
yeah.
yes. I think for any questions still participants have, you can contact sir in mail.
Yeah.
So this is the questions are over, sir. So I am delighted to give thanks to Dr. Lakshman Pulli. On behalf of GPRCP, I would like to express my sincere thanks for accepting our invitation and joining us to enlighten us on the topic of the immunohistochemistry methods and protocols. Sir, with your outstanding presentation, you have filled our brains with new knowledge of that. You started your presentation with what is an immunohistochemistry. That is, even you have explained about the description of antigen antibody reaction and how antigen binds with antibody. And then you have explained about how to make primary and secondary antibodies and application of monoclonal and polyclonal antibodies. So then you have explained about steps involved in immunohistochemistry, that is by special mention to that is of the need of having a prior protocol or planning for the protocol, and the need of having controls, and also explained about how to perform transcardial perfusion and need for transcardial perfusion. And then you have explained about antigen retrieval, that is the universal technique used for antigen retrieval by using sodium citrate. So then you have that is explained about need for blocking endogenous peroxidase and also non-specific binding, especially to increase the S by N ratio, which is very important in the research of immunohistochemistry. And then you have explained about choice of right antibody and how to choose primary and secondary antibodies. And also you have explained about methods of IHC detection, various methods used, and the controls, and also need to have what is the need of having the controls, that is positive and negative controls. So then lastly, you have explained about with your real research that's work, that is image analysis. That is, you have given very detailed description of image analysis and the challenges you have faced, and also that is of in various disease models, especially in Alzheimer's disease, how the image analysis is done. Thank you, sir, for a very nice and informative presentation. So this is okay, sir. So once again, thank you, sir, for sparing the time and sharing the knowledge and your research experience with us. As you said, like we are looking forward to hear you in future and also to have live demonstration and also for research collaborations. Thank you once again, sir.
Yeah, thank you, ma'am. And I'm also thankful to Professor Viresh Mantal, and my greetings to your principal, Professor Madhav Reddy, for providing me this opportunity to present and share my expertise. I'm really happy to have come here and presented my work, and I will be very happy to collaborate and also extend any kind of assistance or be a part of a team which we would like to do collectively. Yeah, and participants are also, they can feel free to contact me if they require any advice or suggestions. Yeah, thank you very much.
I also extend my thanks to our principal, Dr. B. Madhav Reddy, and Dr. P. Prabhashankar, President, IPA Telangana State Branch, MYBO Group, and other organizing committee members for their cooperation in organizing this session. And my special thanks to Mr. Nasheeb Basha, Assistant Professor, and Dr. K. Naresh, Associate Professor, for their technical support in smooth conduct of today's session. So lastly, I thank all the participants for attending and for their patient listening. And I wish you to have a great learning experience for all the days. Thank you once again. Thank you one and all. I hand over the session to Dr. Naresh.
Thank you, madam. Thank you, Lakshman sir, for such an informative session on this immunohistochemistry. Now, I would like to inform all the participants that assessment cum feedback forms are available in our website. Just please visit our website, and it will be activated after 1 hour of this session. And assessment cum feedback link will be active for 24 hours only. Okay, so you can view all the whatever the webinars in our YouTube channel. So if you have any doubts and any queries, feel free to drop a mail at the seminars@gprcp.ac.in. And tomorrow, day 2 of IDDD 2020 e-FDP program, there is a talk by the speaker, Dr. Chandrugoda Rahab Patil, on the topic drug discovery and development in alternative medicine, an intriguing path. So tomorrow we will meet you by 10:30 a.m. Thank you, one and all.